अस्सलाम वालेकुम। अच्छा विजय, हमारा नेक्स्ट टॉपिक है थायराइड ग्लैंड एंड इट्स हार्मोन्स। सो थायराइड ग्लैंड is located immediately below the thyroid cartilage in the neck. As I told you earlier, where the thyroid cartilage is located, it is below the epiglottis. There are many types of cartilage, many cartilages which are there. There is arytenoid, there cricoid, cricothyroid, and then there is thyroid cartilage. It is located just below the thyroid cartilage on each side of the you can say esophagus and the trachea. It is located anterior to the trachea and anterior to the esophagus. Esophagus is the most posterior structure here. The weight of the thyroid gland is 15 to 20 grams in adult, and it is one of the largest endocrine glands. So mostly the endocrine glands are very small in size. Thyroid is a large one. It is present in pair, one on the right side and one on the left side. Then there are smaller glands that are known as parathyroid glands, which are present above the thyroid gland. The thyroid gland is very important for the maintenance of homeostasis in the body, that is the normal functioning of the body. So it is controlled by the thyroid stimulating hormone that is secreted by anterior pituitary. To be specific, it is secreted by the thyrotrophs that are present in the anterior pituitary gland. The signal for the release of thyroid stimulating hormone is produced via thyroid releasing hormone, hormone releasing hormone that is produced by hypothalamus. So this was an overview of the thyroid gland. Now let's look at the other structures which are present here. So the parts of the thyroid gland, there are two lateral lobes, and then there is a part that is known as isthmus. Isthmus is just like a connecting stalk between these two lobes. The posterior side of the thyroid gland is connected to the cricoid cartilage by a ligament which is known as the ligament of Berry. Let's look at the structures which are present here. So this is the right lobe, the left lobe, and in between the lobes there is the isthmus. It appears somewhat like H, capital H alphabet of English, and it is present here in the neck below the thyroid cartilage and anterior to the trachea, and it is also anteriorly located to the cricoid cartilage. There are four smaller glands which are present on the thyroid gland, which are known as the parathyroid glands. Parathyroid glands would release a hormone that is known as parathyroid hormone, and it helps in regulating the calcium and phosphate levels in our body. So what about the thyroid? Let's talk about the thyroid gland. So this is the location of the thyroid gland. Just a second. So we were talking about the thyroid. So as you can see, the thyroid gland here, it is located in front of the trachea. The lateral lobes are located on the right and the left of the trachea, and in between the lobes is the isthmus of the thyroid gland, and it is present just below the thyroid cartilage. It originates from an invagination of the floor of the pharynx and a thyroglossal duct. So in the start there is a ductal structure that is known as a thyroglossal duct, which marks the path of the thyroid gland from the tongue to the neck. So a duct would be present here, and the thyroid would develop on its distal end, and in time this thyroglossal duct disappears. If it is persistent, if it remains in an adult individual, it would be known as a thyroglossal cyst. Let's talk about the functional anatomy of the thyroid. The thyroid gland's blood supply is very unique. It is supplied by two major arteries, that is superior thyroid and the inferior thyroid artery. Superior thyroid artery is a branch of the external carotid artery that supplies the face, and the inferior thyroid artery is a branch of the thyrocervical trunk. That is a major artery that supplies the structures present in the neck region. Sometimes there is an artery which is known as arteria thyroidea, which arises from the brachiocephalic trunk. It acts as an accessory supply to this gland. The venous drainage is same as the arteries. There is a superior thyroid vein, the inferior thyroid vein, and then there is a middle thyroid vein. The superior and the middle thyroid vein would drain into the internal jugular vein that is coming down from the brain, and the inferior thyroid vein drains into the left brachiocephalic vein. So here is the blood supply. The superior thyroid artery is a branch of the external carotid, whereas the inferior thyroid artery is a branch of the brachiocephalic trunk. Then sometimes there is an artery which is known as arteria thyroidea, and it is arising from the brachiocephalic trunk. The inferior thyroid artery is a branch of thyrocervical trunk, which is in turn a branch of the subclavian artery. The venous drainage is shown here. Here is the superior thyroid vein, the middle thyroid vein, and the inferior thyroid vein. Superior and the middle thyroid vein would drain into this internal jugular vein, whereas the inferior thyroid vein would drain into the left brachiocephalic vein directly. If we study thyroid at the tissue level, it has some very unique structures, very important structures. So on tissue level, the histology of histologically the thyroid contains some particular kind of bunch of cells which are surrounded by a membrane, which are known as thyroid follicles. Inside the follicles are the follicular cells. Then there are some other cells which are known as C cells. Follicular cells are in fact the cells which would synthesize the thyroid hormones, that is the triiodothyronine and thyroxine, T3 and T4. The cavity of the follicles would contain these enzymes in colloid form, and moreover it is in this colloid there is a glycoprotein that is known as thyroglobulin. This is in fact the protein by which the thyroid hormone is bound, T3 T4 are bound, and they circulate in the blood via bound to this protein. About 90% of the T3 T4 present in our blood is in bound form, only 10% is free. The parafollicular cells are scattered among all the follicles, and they secrete a hormone that is known as calcitonin. Calcitonin, as signified by its name, plays a very important role in the metabolism uptake of calcium. Basically, the calcitonin would release the calcium from bones, whereas the parathyroid hormone would absorb it in the bones. So these are the thyroid follicles. As you can see here, they are surrounded by the cuboidal epithelial cells, and in between the follicles are the blood vessels, which consist of larger and then smaller arterioles. The T3, T4 and the thyroglobulin is stored inside the follicles in colloid form, and parafollicular cells are present in between the follicles. So remember, whenever there is name of a gland, the epithelium would be cuboidal. So all the glands epithelium, either it is endocrine or exocrine, the epithelium would always be cuboidal. Now let's talk about some hormones that are secreted by the thyroid gland. There are primary hormones which are known as thyroid hormones, which are secreted by the thyroid follicles. These hormones are 2 in number, T3 and T4, or triiodothyronin and thyroxine, or tetraiodothyronin. Triiodothyronin contains 3 of these molecules, thyroid hormone molecules, whereas tetraiodothyronin or thyroxine contains 4 molecules. Moreover, the parafollicular cells of the thyroid would release calcitonin. Now let's talk about the thyroid hormones one by one. The principal hormones which are secreted by the thyroid are the thyroxine and the triiodothyronin. The thyroid hormones would need 2 things to be produced. There is iodine and tyrosine. Tyrosine is the amino acid that would transform after attaching in 3 or 4 molecules into thyroxine and triiodothyronin along with attached iodine. To form normal quantities of T4 or thyroxine, 1 milligram per week of iodides are required. So the patients with low iodine level are at risk of developing goiter. Goiter is a condition where the size of the thyroid is increased exponentially. Common sources of iodine are the iodized table salt that is being used today all over the world. This natural salt comes in smaller packets, and it is very essential for our diet. Moreover, the sea fish and water also replenish the stores of iodine, and all the food grown in iodine-rich soil would contain iodine to much extent, and that would replace the stores in our body. Now, synthesis of thyroid hormone is a very important thing. We'll study it in great detail when we are studying physiology, but now we'll just go through it. So there are many steps that are involved in the synthesis of this thyroid hormone. There is iodide transport or iodide trapping, in which the iodine which is traveling through the blood is trapped and stored in the thyroid. Then there is the thyroglobulin synthesis, oxidation of iodide ion into iodine form, and iodinization of tyrosine, that is addition of iodine to the tyrosine molecule, or organification of the thyroglobulin. Then there is coupling, where these molecules which contain tyrosine as well as thyroid, iodine, are linked together, chained together in 3 or 4 pairs, in the pairs of 3 or 4 per molecule. And then there is the storage of thyroid hormones, which is in form of colloid matter stored within the follicles. Now there's a detail of synthesis of this thyroid hormone. So an active transport mechanism on the basal surface of the thyroid follicle cells pump the iodide within the cell and can raise the concentration of iodine to as much as 250 times that of the blood plasma normally. The thyroglobulin is synthesized in the thyroid gland and it is secreted into follicles. It is a large glycoprotein in nature. Thyroglobulin contains tyrosine amino acid and they are the major substrates, the major parts which combine with iodine to form the thyroid hormone. So thyroglobulin is in fact thyroid hormone coupled with it is thyroglobulin contains tyrosine amino acid and they are major substrates that would combine with iodine to form the thyroid hormone. T3D4's major component is tyrosine and iodine and then linked molecules together. The thyroglobulin molecule provides tyrosine for T3D4 synthesis and store this T3 and T4. The third step in synthesis is the oxidation of iodide to iodine ions, that is, the iodide ions are oxidized to iodine. Oxygen is added to this compound that is then capable of combining directly with the amino acid tyrosine. The oxidation of iodine is promoted by the enzyme peroxidase and its accompanying hydrogen peroxide. So oxygen is added, one oxygen is subtracted from the hydrogen peroxide and water is left. Iodinization of tyrosine or organification of thyroglobulin takes place in follicular lumen. So in the lumen of the follicular cells of thyroid, a portion that is less than 20% of its tyrosine residues are iodinized. The catalyst for this reaction is the peroxidase, and the initial products of iodination are the mono and diiodotyrosine, MIT and DIT respectively. If DIT combines with one MIT molecule, it would form T3, and if DIT combines with DIT, it would form T4. Then the coupling takes place, that is due to peroxidases that promotes the coupling of iodinated tyrosine in the thyroglobulin molecule. When two diiodothyronine are coupled together, they would form thyroxine, that is tetraiodothyronine. When one DIT combines with one MIT, then triiodothyronine or T3 is produced. When iodine is abundant, mainly T4 is formed, but when iodine becomes scarce, that is low, there is more T3 production, so keep this in mind. Then there are the storage of the thyroid hormones. Enough hormone is stored as indicated globulin in the follicular colloid to last the body for 2 to 3 months. So the thyroid gland would release T3 or T4 from the follicular lumen, which would contain the stored form of T3, T4. Cuboidal storage of this iodine as it forms T3, T4 is given here. Okay, bache, I think this is enough. I'm actually not feeling very great today, so I'll record more and combine the lecture tomorrow in the morning and then upload that. So there'll be only one lecture on Friday. Assalamu alaikum. So we were talking about iodide transportation, trapping, and thyroglobulin synthesis. So what happens is iodide is basically an ion form of iodine. When this iodide is combined with tyrosine, that is an amino acid, and makes up thyroglobulin. Okay, so and they would form the T3 T4, that is the enzyme, primary enzyme that is produced by the thyroid gland. So what happens is oxidation of iodide ions takes place. When oxygen is added to the iodide, it would change into iodine, and then oxidation means addition of oxygen or removal of hydrogen. So what would happen is this oxidation occurs, and this oxidation occurs under the effect of enzyme peroxidase and hydrogen peroxide. Now then organification of thyroglobulin occurs. That is iodine is attached with tyrosine and it forms MIT or DIT. Thyroglobulin is extruded into the follicular lumen, and less than 20% of its tyrosine residues are iodinated. The catalyst for this reaction is peroxidase enzyme, same which was used in the last step. Here you can see a tyrosine molecule. This is monoiodotyrosine, that is one iodide is added, and diiodotyrosine where two iodides are added. This is the iodine, the red line. One is added here, two are added in this structure. Now this mono and diiodotyrosine would couple together, they would mix up and produce T3 or T4. If mono MIT and DIT combine together, they would produce T3, and if two DITs combine together, they would produce T4. So this is also promoted by the peroxidase enzyme. Peroxidase would promote the coupling of iodinated tyrosine in the thyroglobulin molecule. When two DITs couple, tetraiodothyronine or T4 is formed, or thyroxine is formed. When one DIT and one MIT combine, triiodothyronine or T3 is formed. When iodine is abundant, the body has enough iodine, mainly T4 is formed. But when the iodine becomes scarce, that is low concentration of iodine in the blood, then the production of T3 would increase. So this is the storage of hormones. The thyroid hormones, they would be stored in the follicular form in the thyroid inside the follicular lumen. They are stored combined with the thyroglobulin, and they can last for 2 to 3 months. So how are they stored? The MIT DIT ions and T3 T4 are coupled together in the form of thyroglobulin, and inside the it is happening inside the follicular cell. How is it released? There are various steps which are involved in the synthesis and then release of the thyroid hormone. There is endocytosis, fusion, proteolysis of thyroglobulin, secretion and deiodination. Endocytosis is whenever a cell ingests another structure, that is known as endocytosis. So pieces of the follicular colloid are taken back into the follicle by endocytosis. So follicle basically eats up the pieces of follicular colloid. Then the fusion would take place. The endocytosed material fuses with lysosomes. Lysosomes are enzymes which break down things inside the cell. Organelles. Lysosomes are small organelles which contain lysozymes which would break down different substances. So these lysosomes which would transport it toward the basal surface of the cell, that is the base of the cell. Then proteolysis of thyroglobulin would occur. Thyroglobulin is a protein. It would get broken down. So the lysosomes with their lysozymes which are present inside these organelles would break down thyroglobulin into T4, T3, DIT and MIT. So thyroglobulin is basically a long chain that contains MIT, T3, T4, DIT, etc. But it is broken down by lysozymes inside the lysosomes in the thyroid. Then the secretion of T3 and T4 would occur into the blood. This secretion would be directly into the bloodstream, and the ratio would be about 20 to 1. So 20 molecules of T4 to 1 molecule of T3. Then the deiodination would occur, that is, iodine would be removed from the unused DITs and MITs and it would be reutilized again. So deiodinase enzyme would remove the iodine from the iodinated tyrosines, that is DIT and MIT, diiodothyronine and monoiodothyronine, but not from the T3 and T4. The iodine is then available for resynthesis of the thyroid hormone. So this is how it goes. The thyroglobulin in the colloid would be ingested. This is the colloid, this is the follicular cell. So this is the lysosome. A lysosome would endocytose the thyroglobulin. The follicular cell would endocytose the thyroglobulin. Then the lysosome would act here and proteolysis of the thyroglobulin would occur. Deiodination would occur. T3, T4 would be secreted outside the thyroid into the bloodstream, and tyrosine and monoiodotyrosine and diiodotyrosine would be deiodinized. Tyrosine would break down and iodide ions would be produced separately. These iodides would be reused and tyrosine would be reused too. How is it transported in the blood? So basically T3 and T4 are lipid soluble hormones. They cannot be transported freely. That's why they are bound to plasma proteins. These plasma proteins are known as thyroxine binding globulin. T4 has a higher affinity for binding proteins, therefore it binds more tightly to the TBG than T3. Consequently, the half-life of T4 is greater than that of the T3 molecule. So thyroid binding globulin would bind T3 and T4, that is more than 99% of the T3 T4 would be bound form, and less than 1% would be in free form circulating within the blood. Now, when it reaches its potential target, it would bind with the same nuclear receptor. T3 and T4, they bind with the same nuclear receptor, but T3 binds more strongly than the T4. So T4 would bind more strongly with the thyroxine binding globulin, whereas T3 would bind more strongly with the receptor on the end organ. Thus, because it has greater affinity for the receptor, T3 is more active form of thyroid hormone, whereas T4 is more kind of dormant form which would just combine with the TBG and circulate within the blood. Hence, most of the T4 converted to T3 at the tissue level. So when it reaches the tissue, T4 is again deiodinized, one iodine is removed and it is converted into T3, and this T3 is used up by the tissues. So remember this from MCQ point of view, T3 is more active hormone and T4 is biologically less active. So thyroxine is activated via an enzyme that is known as 5-monodeiodinase, that would remove one iodine, and this T3 would be reused. Whereas degradation occurs also in the same pathway. Now let's look at some differences between the T3 and the T4. So what are the differences? T3 contains one molecule of MIT and one molecule of DIT. So 2 plus 1 equals 3. Whereas T4 contains two molecules of DIT, that is 2 plus 2 equals 4. Plasma concentration of T3 is less, whereas plasma concentration of T4 is more. As we've already studied, the plasma ratio is 20 to 1. 20 T4 molecules, 1 T3 molecule. Whereas T3 has lesser affinity for binding proteins. It tends to move freely, but still more than 90% is bound with the thyroxine binding globally. T4 has higher affinity for binding proteins. It is mostly in bound form. Whereas T3 is more active form of thyroid hormone. It would more strongly bind with the receptors, whereas T4 would not bind so strongly, and it is a less active form of thyroid hormone. There won't be any conversion of the T3, it would be just used up and degraded. Whereas T4 is converted into T3 at tissue level to be used. The mechanism of action, we'll study this in the next lecture inshallah. And if you have any questions, please post it in the WeChat group. Okay, don't post the questions or don't chat in the Ding Talk group. The Ding Talk group, it is only for marking your presence. Okay, so don't use it for unnecessary messages. Thank you very much.